So hello, everyone. I am Douglas Mans, Environmental Molecular Sciences Division and Environmental Molecular Sciences Laboratory User Program Director. And as Brian mentioned, I started last April in 2019. I thought I would start maybe with a little preamble. Some of you may know, but I realize many of you may not yet know that EMSL, the Environmental Molecular Sciences Laboratory, is a DOE Office of Science user facility funded by the Office of Biological and Environmental Research, also known as BER. You may hear their name from time to time in this presentation. As a user facility, we fund competitively peer-reviewed projects that further BER research from scientists around the world, but we are also a P&L division. Though most of our funding comes from BER, we also work on projects funded by NIH, NSF, and other sponsors. To support the user program for BER, the EMSL facility houses our scientific expertise, instruments, and unique laboratory spaces within the EMSL building. Briefly, the facility encompasses a total of 234,000 square feet, where we house and operate over 150 unique instruments. In addition, there are several unique laboratory spaces in EMSL. This includes our prime computing space for Tahoma, our newest high-performance mixed hybrid architecture supercomputer that's replacing our aging Cascade system. We have a quiet wing that is acoustically, vibrationally, and electromagnetically shielded with 8 bays housing electron microscopes, a plant science laboratory with controlled growth chambers, phytotrons, and various root and rhizosphere imaging capabilities, and a highly modified lab space that houses our 21 Tesla Fourier transform ion cyclone resonance mass spectrometer, which is one of 2 in the world, the other being at the High Field Magnet Lab at Florida State. For FY20, the user program supported 685 users on 201 active projects with an operating budget of just under $45 million, and we're anticipating a similar level budget for FY21. So I thought it would only be appropriate to kind of set the scene with the agenda on a talk about roadmaps with a roadmap to orient where we're going to go. And so the talk today will be largely chunked into 3 portions: where we are now, our current state, where we want to go, and our aspirational future state, and how we plan to get there with our S&T tenure objectives and roadmap. So if I start with where we're at, being a BER user facility, our current mission, directions, and science focus are inextricably tied to and influenced by BER science mission and focus. To that end, our vision for EMSL is to provide a predictive understanding of dynamic molecular transformations underpinning critical biological and ecosystem functions. And our mission is to lead the development of and provide access to multidisciplinary characterizations of in situ dynamic molecular transformations, establishing functional knowledge across scales for biological and environmental systems. Focusing on our mission then, the leadership team and I have crafted 2 overarching aspirational goals that EMSL and the division are looking to pursue in the future. Our first goal is to be premier science leaders in our 2 science areas aligned to supporting the BER science mission. The 2 are represented pictorially here as functional and systems biology on the left and environmental transformations and interactions science area on the right. These 2 areas are meant to be the avenues for progressing BER science in the user program with users, as well as creating an area of focus for where our science leadership will continue to be enhanced going forward. Our second goal is focused on addressing 2 challenges associated with BER research that EMSL currently is facing. At its core, BER science is big science. This requires a multidisciplinary expertise where increasingly more and more cross-cutting questions are being asked, generating more and more data and knowledge, which creates this virtuous cycle in which more disciplines end up being sucked in as knowledge is shedding light in new areas, creating more new questions. At its heart, EMSL represents these big science capabilities for BER with our facilities and the breadth of instruments and expertise housed within. However, there are 2 challenges that we face as we look to enhance our leadership in multidisciplinary science. The first is how do we maintain leadership in developing cutting-edge instrumentation and applying it to new areas of science of interest to BER as the barrier to entry for acquiring high-tech instrumentation drops. It's increasingly more common for even modestly funded labs, individual researchers, and institutions to acquire and utilize many of the same high-end instruments that once were the domain of specialized facilities like EMSL, as the advances in the instrumentation are rapidly commoditized. And secondly, simultaneously as a user facility, we need to ensure that EMSL can provide the capacity at the pace BER and more broadly DOE science requires. Two examples of this are that, right now we've reached essentially a tipping point where the velocity of genomics data generation is outstripping our ability to accurately and efficiently convert that information into functional knowledge of biological systems. Additionally, our Earth system models have begun approaching the limits of resolution and accuracy and will require a wealth of new molecular level data and parameters to provide that next level of accuracy and resolution, which is needed to continue enhancing our model's predictive powers. Both areas in BER require a capacity and a pace for data generation and data analyses, leading to knowledge that is a current gap in DOE capabilities that EMSL is positioned to address. As a result, our second goal we're pursuing is to simultaneously pursue cutting edge science that provides greatly enhanced capacity and pace to maximize BER scientific impact. Our strategy to accomplish our goals is focused on a multidisciplinary platform approach to create domain expertise in 6 core areas, shown on the right side of the screen, referred to as our integrated research platforms, or IRPs. These IRPs contain our diverse science expertise, our advanced instrumentation, and often unique facilities to foster domain expertise in these areas. These platforms can serve as standalone areas of focused research to advance those science associated with these IRPs. But more excitingly is the opportunity to sort of mix and match these IRPs like Legos to leverage synergies and amplify science impact at the interfaces. Computing, analytics, and modeling was viewed as so foundational to all of the IRPs and our science areas that we created it as a standalone group, which is represented along the bottom of the screen on the slide that you see. So armed with our vision, our mission, and our goals for EMSL, along with a strategy focused on IRPs to align our activities against, our next task was to more clearly define where we want to go in pursuit of our 2 goals. What does science leadership in our 2 science areas look like or mean in the future? Starting with our first science area in the future, as science leaders in functional and systems biology, our intent is to focus on being able to predict and control biology for sustainable energy and ecosystems. The intent is to move the science community to a post-genomics world, if you will, where biological function is determined through deep knowledge of metabolic and systems biology, as opposed to merely relying on homology or association. This would allow us to rapidly and accurately decipher a protein's function, or perhaps more appropriately, its functions, given that they often have multiple roles dependent on the context. And more importantly, allow us to completely understand how those functions are affected by and affect other proteins when placed in the context of complex metabolic pathways. How perturbing these interacting pathways at the genomic, and perhaps more importantly at the environmental level, ultimately affect function and physical traits at the cellular, communal, and plant systems level. Our goal is to use this deep understanding of phenotype to drive design of systems for bioproducts production and enhance resilience properties of ecosystems. In the environmental transformations and interaction science area, it's about following and deciphering what I refer to as the molecular Twitterverse to predict ecosystem response. I liken the concept to that of Twitter, where someone has a local network of friends to whom they send out these small 140 character tweets to convey or elicit some type of response. Similarly, the microbial, soil, plant, and atmosphere systems create these small local network of friends in which small molecules are constantly being sent out and received by microbes and plants and are being acted upon by the soil and the atmosphere. These molecules cause a response, which is then communicated back out via another round of molecular tweets. However, this is where it gets interesting. In the social media world, your friends likely have friends outside your network. And so when one ends up with these interconnected networks of networks, in which tweets from one network get transmitted across into other networks, and the responses end up rippling across a city, a nation, and even globally. Just as in the world of Twitter, these local plant microbe networks also create this network of networks in which the mycorrhizae, for example, transmit molecular tweets terrestrially among plants and microbial communities, and the plants also release volatiles via the atmosphere. If we can more completely decipher what these molecular tweets mean and how they lead to responses, you can build up a predictive picture. If we recall the Arab Spring in 2010 in the Middle East, Twitter was used as a very effective communication tool to coordinate activities across countries. Mining the tweets and deciphering their content allowed us to build a predictive model of where and when social unrest was most likely to occur. Our goal is to accomplish similar predictive modeling capabilities on an ecosystem scale. So to move EMSL as premier leaders in our science areas and realize our two overarching goals, we embarked on an effort to create a set of strategic tenure objectives directly supporting these goals that would be transformative for BER and squarely position EMSL as leaders in our science areas. To craft these objectives, we hosted a series of 3 road mapping workshops from February to June this past year. The workshop participants included science and technology leaders from across PNNL, EMSL, and the external science community. And during these workshops, we assessed current scientific trends and drivers, sponsor needs, as well as national and societal priorities. Based on where we saw the future of BER research heading and the larger trends and drivers that impact EMSL, we formulated the following 3 strategic tenure objectives. The first is creating a digital phenome, or DigiPhen as it's referred to, as a platform for predicting and controlling biology from biomolecules to organisms. The second is establishing MONET, a national molecular observations network for modeling from elements to ecosystems. And the third was building a BER-focused modeling and data sciences capability to visualize all BER's data. So having crafted our 10-year end state of what EMSL looks to provide as premier science leaders for the BER research community, the second and third workshops, along with a lot of offline virtual meetings due to COVID, focused on building a more granular view of the specific activities and advancements referred to as value streams that will be required to achieve our 3 objectives. This became the plan for how we will get there, or essentially our S&T roadmap. So you may ask, so what does a roadmap look like? And the reality is we actually have multiple roadmaps. We have one roadmap for each objective, and then several underlying roadmaps in support of how we're going to do the various activities associated with the objective roadmaps. So starting with our first objective then, we endeavor by 2030 to establish a digital phenome platform for predictive modeling of protein function, pathway identification, and phenotype. There is a need to produce higher throughput and robust protein function characterization combined with an understanding of complex metabolic and regulatory pathways spatially and temporally to unlock critical insights needed for controlled engineering of biological systems for bioproducts production and to address environmental challenges. With this objective, EMSL will lead the effort to develop a platform based on data model integration across multiomics, structural biology, and cellular signaling. DigiPhen will dramatically increase the accuracy of and accelerate the association of genes with phenotypes, identifying wholly new protein and metabolic functions, and enable model-driven engineering of plant, fungal, and microbial physical traits critical for scalable bioproducts synthesis, as well as enhanced ecosystem resilience and feedstock sources. Accomplishing this first big, hairy, audacious goal, or as it's referred to in the entrepreneurial environment, these BHAGs, will take the combined effort of our internal expertise, our world-class instruments, and our remarkable facilities. Unfortunately, I don't have the time now to delve into all of the timeline specifics for all of the associated activities, but I will comment that large aspects of the research look to enhance on our already strong leadership role in functional omics through high-put omics and protein function, visual proteomics, and visualization of metabolic pathways. In addition, there are a few opportunities where we felt we could chart new capabilities in single-cell biology, enzyme site active understanding, and bioatomic imaging. You also note a large aspect of automation and standardization associated with many of these activities, which will bring the capacity and the pace needed as well. One area that I think is extremely interesting looks to embrace more fully the open science movement to enable EMSL and the BER community to scale our ability to probe biology for function. Loosely termed the Molecular Biology Institute and some associated molecular biology satellites, these activities look to use small molecules to probe biological function. By using automation, standard chemical probe protocols, and metadata schema, we're envisioning scaling out the probing of thousands of proteins by creating EMSL molecular biology satellites, where workflows and even automated platforms could be placed at institutions across the US, each looking at BER relevant proteins and systems, but at a scale unrivaled and that which EMSL itself could not accomplish. All this data on the biological function could then be assimilated back into EMSL's database, Nexus database, to create a massive library of mineable content to create connections and wholly new insights about metabolic function across species and kingdoms. For our second objective, by 2030, we're looking to establish MONET, a national molecular level watershed, coastal, and terrestrial observations network. There's a need to fully understand ecosystem dynamics through a more complete data model integration. and EMSL sees an opportunity to develop a national network of remote sensing and observing capabilities that will be coupled with standardized sampling and high throughput automated analyses capabilities. This network will provide molecular level understanding of ecosystem perturbations and health to enable predictive modeling. Ultimately, MONET will allow for scientifically informed decision-making regarding energy and water sustainability for the US, as well as provide more accurate Earth system modeling capability through integration of the appropriate molecular and mechanistic level interactions. Broken into 3 phases in this objective roadmap, Phase 1 of MONET looks to supercharge the foundational work on watersheds that is being pioneered in the WONDERS program led by James Stegan and collaborators here at PNNL. And then link in and as appropriate, support the coastal pilot, which PNNL is also spearheading, supporting and providing molecular level observations and sensing in these marine environments. Phase 2 then looks to extend and connect with by growing multi-agency partnerships into terrestrial space, including, for example, Spruce, CZO, NEON, and USDA research sites. Phase 3 of this would then complete the network through the tying of the ecosystem networks together in a holistic manner at a national or continental level. I think there's lots of really cool opportunities in this roadmap specifically, especially when combined with the 5G and beyond capabilities for remote sensing and equipment operation, as well as the EMSL labs of the future that we're planning. For example, you'll notice in the near term, there's a large focused effort on complete automation and standardization and sampling workflows for dissolved soil and natural organic matter analyses. In addition, we'll be focusing on new sensor and observational developments, which are looking to leverage unique control and compute capabilities that 5G and other wireless connectivity would usher in for field deployment. At the very bottom, you'll notice there's an experimental and computational linkages that are of high interest to BER and DOE, especially in the artificial intelligence and machine learning space, in the operation of instrumentation, and very importantly, as well as in the model-driven experimentation and data model integration approach to drive experimental design and directions based on continuous monitoring and analyses events. And the last point I'd make on this one is I see tremendous opportunities to forge some amazing partnerships in the open science perspective with other facilities, independent researchers, and government agencies. Our third objective is to build by 2030 a BER-focused modeling and data sciences capability, which would provide mid-range high-performance computing capabilities for data model integration and multi-scale modeling across the BER science portfolio, spanning the molecular mechanistic level models up to climate and Earth scale simulations. The intent is to leverage existing capabilities that BER already funds to more broadly support BER's mission. To do this without compromising EMSL's operation, the intent is to pursue BER funding alongside EMSL funding to sustain these capabilities. EMSL, the facility, the actual building, has about 8,500 square feet of prime computing space and a staff of world-class high-performance computing and data scientists that can be leveraged to provide the mid-range computing capabilities for modeling and data science work that is currently a bottleneck for BER within the Oscar space. In this area, EMSL is already a major player for BER. For example, in the metadata capture and fair data management area, we're already actively helping define fair metadata schema as part of BER's new National Microbiome Database Collaborative initiative, which we are an AIM lead and significant contributor of. In addition, in the open source data analysis and software suite, our historical and continued development of the open source modeling tool NWChem for biological and environmental applications, along with IPMART and a variety of other metabolic visualization software suites are prime examples of our play in this space. However, there are also several new areas that were highlighted as strategic offerings that EMSL could lead, for example, including the imaging processing capabilities associated with cryo-EM, X-ray computed tomography, and light source experiments. Having worked through how to do this with our role as the co-lead in the Pacific Northwest Cryo-EM Center in partnership with OHSU, as well as recently bringing online our own Titan Cryo-EM, as well as 2 X-ray computed tomography units. And as I mentioned earlier, the computing space in EMSL is sufficient that we could also look to pursue cutting edge development and deployment of artificial and machine learning applications for automation, ideally in partnership with Oscar. I mentioned it briefly in previous slides regarding EMSL's vision for embracing 5G capabilities and automated practices. Thankfully, automation was a journey EMSL was already embarking on prior to my arrival. but COVID and the need for business continuity in midst of shutdowns has merely served to accelerate our planning for EMSL's labs of the future, which are required to achieve the goal for dramatically enhancing capacity and pace of research, as well as to secure our role as science leaders. Envisioning this lab of the future, we are endeavoring to pursue fully automated modular workflows with minimal human or no human intervention, where non-standard, hard to automate manipulations, i.e. lab settings, are actually automated. And to do this, we'll be looking to leverage AI and machine learning supported analyses and decision informing tools for model-driven experimentation-focused operation. Ideally, these would lead to 24/7, 365 autonomous and remote operation enabled via web-based interface for researcher and operator control. And these workflows would be self-contained in order to maximize efficiency, minimize cross-contamination, and enhance researcher safety. And as you can imagine, as the 5G wireless advances, as well as the Internet of Things advances continue to transform how we connect various platforms, you can envision a situation where we can now start to update platforms via Tesla over-the-air type updates, which would facilitate the satellite operations I mentioned earlier, part of this so-called scale out versus scale up approach to supporting BER science at a larger capacity and pace. And so some very preliminary thinking for future EMSL facilities and potential for infrastructure investments are looking to be focused on an automation wing. Within this facility would be contained fully self-contained pods, nominally 10 foot by 10 foot by 20 foot, but you can envision various sizes of these pods, but they would be dedicated analytical workflows per pod. And this would be a very efficient use of the space with minimal infrastructure required. And the modular design allows for a very rapid deployment of new capabilities, as well as a mixing and matching of analytical workflows to create potentially whole new workflows. And in this manner, operational costs are minimized while we maximize scientific productivity through high throughput capacity and pace for DOE science. Taking this to the extreme, one could then envision these pods to become deployable, at which point we could then envision taking these to field sites, similar to the atmospheric radiation measurement facility, our ARM, which is another user facility that PNNL stewards, or to place these at satellite institutes with 5G connectivity or other wireless connectivity back to EMSL's Tahoma and Nexus systems for data analysis, mining, and archiving. So I mentioned a bit about EMSL's labs of the future and the need to embrace multidisciplinary expertise that's going to be required. Our facilities are going to need to enable and support a makerspace culture where we remain flexible and fluid enough to seize on exciting new science opportunities, but at the same time, facilitate the operation of automated high throughput workflows to support the pace and the capacity needed of EMSL's cutting edge techniques. From a facilities perspective, we have three unique options to consider individually, or more likely in some combination in the very near future. The first, as many of you are aware of, is the energy sciences capability move that will open existing space in EMSL. As I alluded to on previous slides, we also have prime computing space that's available as a result of continued hardware miniaturization. And third, we have an area north of EMSL that's available for expansion to house new or increased capabilities. As such, an EMSL facility steering committee has been created to prepare options for a set of near-term space occupancy needs with the ESC move, and the longer-term plan for a more holistic layout of EMSL aligned to a set of guiding principles aligned to IRPs that would be transformative to EMSL and the EMSL user facility model in general. For each of these spaces, we have already identified a subset of specific investments options that we have begun to pursue. Two of the more exciting near-term space plans include the creation of a dedicated makerspace, as we call it, to enable cutting-edge science to be matched with creative technology development across our integrated research platforms. As I alluded to on numerous other slides, automation will be a core focus for EMSL's operations of our platforms for 24/7, 365 access to users. As a first pilot for this, we've identified and have been working with the WONDERS program, led by James Deegan and collaborators, to build and deploy a fully automated dissolved organic matter analyses workflow, starting from standardized pre-barcoded or otherwise labeled sample collection kits and metadata capture to sample pre-treatment, carbon analysis, preparations, and loading into our FTICR mass spec for analyses, and data processing and upload to the EMSL Nexus database. So this is all with no human intervention. So the samples come in at the dock and they get disposed of in the waste bin in a completely automated manner with hopefully zero human intervention. To facilitate this, a suite of labs to be vacated in the ESC move have been selected to house the workflow, as well as serve as a test facility to map out more fully specifications and requirements for more broadly automating EMSL's workflows as part of our future planning. So over the last about 25 minutes or so, I've spent a great deal discussing EMSL, our capabilities, and our future. Although EMSL has tremendous breadth of experience and expertise and capabilities, the truth is we cannot, nor should we, do it all. We recognize that for us to make significant progress on our tenure objectives for the benefit of BER and to provide the premier science capabilities required by the science community, we need to partner and leverage synergistic capabilities of other user facilities and even agencies. As part of our operational strategy, EMSL is looking to broaden and create a user facility network where EMSL could be considered a hub given our multi-purpose focus for BER. We already have a very successful and long-standing partnership with the Joint Genome Institute through our FICUS program that enables users access to both facilities through a single proposal. We've also partnered with ARM in a FICUS manner to combine our sampling and molecular characterization capabilities with the aerial and deployed atmospheric capabilities at the ARM facility to better understand aerosol composition and fate. In addition, this year, we have a small pilot partnership with NEON, the National Ecological Observation Network that's operated by NSF, to conduct a prototype continental-based molecular level microbiome studies using their field site sample library that's combined with the EMSL and JGI multi-omics and organic matter data libraries that we have. In addition, we have some very promising discussions going on now, engaging the spatial neutron source and the high flux reactor at Oak Ridge to use their novel isotopically labeled imaging capabilities associated with the biosams. As well as we've been having some preliminary discussions with the advanced photon source at Argonne to leverage the upgraded capabilities as part of the APSU and its structural biology and molecular imaging of minerals and soils capabilities. So I hope that our 10-year strategic objectives not only further BER science in pursuit of sustainable energy sources and environmental security, but we hope that we can do our part to reinforce the importance of interdisciplinary collaboration in answering the many scientific grand challenges we will face for many years to come.